what do antibodies do? They bind antigens. Now, the antigens are supposed to be what in your body? What do the antibodies bind if it's like in your body? It's an antigen, but what does that really mean? It's a foreign body. It's something that's not supposed to be there. Okay? So the antibodies that are roaming around in the bloodstream can bind to it and cover it. Okay? And since it's in an animal system, it's polyclonal. That means it'll bind it all over the place in different spots. And one thing that the antibodies do is by covering it, it prevents that foreign body from interacting with anything else. So effectively makes that not functional anymore. We even now use antibodies as a drug so that the antibody will bind to whatever we're trying to inhibit, and it won't be able to interact with the natural things that it wants to interact with. Okay? The second thing the antibody does is it will tag that entity to be destroyed. If it is a small thing, then the fact that antibodies are covering it, it means all of the constant regions, or the FC regions, are sticking outside. That is an indication for a macrophage to bind it, internalize it, and break down whatever's there. Once you've broken something down, whether it's the DNA, the RNA, or the protein, you've destroyed its function, you've destroyed its information. If by chance the antibody binds to something too large, like a cell that can't be ingested, we have another way or mechanism in such a way that we have what's called the complement fixation, in which we have a series of proteins interact amongst themselves and insert proteins into the membrane to create holes, and the cell will be destroyed, broken apart. And then all the little pieces then can be covered. Okay. Also, the beta cells have the same binding pockets as the antibodies they create. So if a beta cell binds to something, that will cause the beta cell that produces antibodies to, one, reproduce, create memory cells, and two, make more antibodies. Okay, and it's a race of time between how fast the invading body, like a virus, goes in and invades your cells versus how fast your immunity can destroy the things. Usually one wins out for a little while, and then hopefully your immunity catches up and overtakes it, and you are, all the virus and all the material is gotten rid of in your system. All right, so that's antibodies and beta cells. But there's a whole different mechanism that we have and we place in the cells to check to see, is my cell okay or is my cell not okay? Okay, and that's what we call the MHCs, and we have class 1 and class 2. So we started looking at class 2. Class 2 presenters, okay, that's what we're really looking at, are incorporated in macrophages. Remember, the antibodies covered something bad, the macrophage took it in and broke it down in the correct way. So it encountered a virus, but it wasn't invaded by that virus. The macrophage took care of it. All right? There is a mechanism as it breaks down things to take a segment of whatever it breaks down and takes one of the peptides, a piece of the peptide, okay, of the foreign agent, whatever is in the body, and it puts it into the MHC, which is this guy right here. That's the MHC. Here's the peptide, all right, and it presents it on the outside. Now the MHC2s bind to receptors that are on the helper T cell. Okay. And so the helper T cells, the receptor on the helper T cell and the MHC2 naturally like to come together and bind. But they have a very weak binding. And so if a helper T cell goes to a macrophage and it binds to an MHC and there's nothing there, no peptide present, it binds transiently and then falls off. And if it binds to one that does have a peptide there, what happens is the binding is a little bit tighter. Because the MHC is presenting a protein piece that was a foreign invader, okay? And this helper T cell recognizes there's something there. And it binds a little bit tighter, tight enough that this protein, the CD4, is able to clamp things down. The receptor on the helper cell and the presenter on the macrophage usually bind so quickly and let go so quickly, C4 can't get in and clamp it all together. It will only allow that interaction to be tight enough if there's a foreign peptide, and then the CD4 clamps together. When you have that clamp, you have the presenter, the foreign peptide, the receptor, and the CD4 all come together. It causes conformational changes of the proteins on the inside of the helper T cell, which causes an event of things that occur. And what happens are interleukins are released. The interleukins will do a couple things. All right? They principally will cause beta cells to be produced. And the beta cells it produces are the ones that are a match to this peptide. So the helper T cells say, hey, we're seeing the macrophages are encountering something. We need more antibodies. This reinforces that I need more antibodies. That's what helper T cells do. They will also stimulate other helper T cells to get working, and they will stimulate the cytotoxic T cells or the killer T cells to work because we might need those working too. Okay? And so the macrophage has encountered a virus but has broken it down, and the macrophage is still healthy, functional, and doing what it's supposed to do. All right? Class 1 MHCs are found on almost all cells in the body. Class 1 have a whole different purpose. All right? How do I know if the cell is good and functioning properly or if the cell's infected? Well, if you've got a membrane on the outside, that membrane looks like everybody else's membrane. But you're invaded and you are changed at the DNA level with a virus coming in. Well, how in the world do I know if that cell is good or bad? Well, if a cell is good, it produces proteins. If it is bad, it produces proteins. Some original and some viral proteins. If it is good and healthy, proteins live a life, do its function, retire, and then get recycled. Okay? So proteins are always being made and always being broken down. In the normal process, the MHC1 class always takes a sample of what's being broken down. And it'll bind a little piece of protein and it'll present it on the outside. Now, the MHC1s are always presenting pieces of protein on the outside. But most of the time, and if it is healthy, it's presenting normal pieces of protein that are supposed to be there. The cytotoxic T cells, or the killer T cells, will come and bind, and if it sees the protein that it recognizes being self, it'll float away. If by chance your cell has been infected, it's producing normal proteins, but it's also producing viral proteins. Through that cycle of making and breaking down proteins to the normal, it will randomly pick out some of the virus protein and put it on the outside. All right, so it'll have normals in most of them, but maybe a piece of viral protein that wasn't supposed to be there. The cytotoxic C cell will come and bind to it. If it's normal, it falls off. If it is foreign, it binds a little bit tighter. Okay? And so the cell here is represented as a virus-infected cell. It's producing viral proteins. It's repackaging, but through that process, the MHCs are always presenting what kind of proteins are in the inside. And so we take all proteins, go through a life cycle and get broken down and recycled. they get packaged to the MHC1s and are presented to the outside. Okay. Most of the time they're normal, but every so often there's a foreign. The MHC class has a binding pocket right here that I'm going to put a peptide that sits right there. And it gets presented on the outside of all cells. You can actually see the binding pocket. It is one alpha helix, and then this place where you sit the peptide. And you can actually see, here's the peptide between the two alpha helix. Here's an alpha helix, there's an alpha helix, there's the beta sheet. It just sits in this pocket, binds very tightly. Space filling model. Here is the protein that is being presented from the inside of the cell, and it's just sitting out there, presenting. The cytotoxic T cell will bind. If it is normal protein, it lets go and floats away to another. But if it's a foreign one, the MHC1 and the receptor on the T cell binds tightly, and I get another clamp. It's not CD4 this time, it's CD8, because it's a different protein, different cell. The CD8 then clamps everything together. I get a change in the function inside the T cell, and you usually also have interleukin 2s, which were coming from helper T cells that are helping the signal. What happens is, once this is bound, the killer T cell initiates apoptosis in this cell, and we kill it. Once a cell is infected by a virus, it cannot be corrected, it cannot be cured, it can only be destroyed so that it doesn't infect other cells. And so here's the killer T cell, here is the MHC1, there's the foreign peptide. If it's foreign, it binds a little bit tighter, allow CD8 to come up and clamp it down and cause this complex. This is the complex that initiates the target cell being destroyed. Yes? Yeah, what's the difference between CD4 and CD8? CD4 proteins are in the helper T cells, and CD8 proteins are in cytotoxic C cells. They are very similar in function and very similar in shape, but they are found in two different cell types. And there was a animation with some sound, but I didn't hear nothing. Let's try this again. Okay. Can't hear it very well, but it's just showing you that when the killer T cells or the cytotoxic T cells bind, it will send out small molecule signals that initiate apoptosis. Now, what's interesting about this and the animation that's showing here is that the killer T cells can invade and identify viral infected cells, but also the T cells and the cytotoxic T cells also can recognize cancer cells. And so cancerous cells are cells that just grow uncontrollably, unregulated, and so you may have cancer cells roaming around in your body every so often that don't form into a cancer because our immunity actually is fighting against it all the time. And so this, because of mutations that can occur in nature, the cytotoxic T cells actually work in that mechanism, and that's where the animation came from. Yes? Apoptosis is programmed cell death. Okay, and so it's a fancy word for the cell is killed.
Did you say that cancer cells are killed in the class 1 MHC?
In terms of yeah, in terms of class 1s, yes. And so presenting through class 1s, you can actually see some of that. Because what you will do is a cancer cell will sometimes create a protein that is different than the normal. And so you can present that part of the protein that is different. Now, here is why we need to look at this when we do transplants, okay? And we segregate these in two different classes. The class 2 proteins are taking a piece of protein from the inside of the cell and presenting on the outside. Class 1s take proteins from the inside and present it on the outside of cells. But class 1 is representing all proteins inside the cell, okay? And so whatever's inside that cell. And so if there's a foreign protein inside your cell from the class 1, that means it's roaming around doing whatever it wants to do, and you're infected. The class 2 is different. The class 2 peptides only come from the mechanism in which we've ingested something. We have broken it down in a normal fashion. We have taken the foreign agent and we have made sure that it is now inert, and we present that on the cell surface. One is presenting a foreign peptide in a mechanism in which I have taken care of it. The other presents a foreign peptide that I didn't even know it was in there, and I'm infected. And so for class 2, I need help, the helper T cells. For class 1, that cell is infected. I need to kill it. I need the cytotoxic T cells or the killer T cells. Yes?
So if a cell has the class 1 MHC signal, will it also have the class 2 signal?
No. Class 2s are in macrophages, okay? Because that's where the mechanism in which we take care of and destroy foreign entities. All cells have class 1. So macrophages have class 1 and class 2, but all other cells only have class 1s. Okay? And so we have the class 2 in the macrophages. Yes?
How does, you mentioned, I think, when you made something about, like, organ transplant?
Yes.
How does this...
Okay, so right here. Here's the problem with organ transplants. All right? On average, each individual has 6 class 1 and 12 class 2 proteins. But there are lots and lots of different versions of this. And so, here's the deal. Especially in class 1 presenters. If by chance you have 6 different versions from me, okay? Inside the cell, inside the red blood cell, there's hemoglobin, okay? And hemoglobin gets broken down all the time. My 6 class 1s will take the C terminus and this segment and this peptide and present it, okay? And so I present a normal protein, hemoglobin, in these 6 different ways. But if you had 6 different MHC1s, okay, then what you would do is you might take the N-terminus of hemoglobin. It's a perfectly normal protein, and you may take other portions of that and present it on the outside. If by chance your tissue gets into my body, even though it's presenting a normal hemoglobin, it's presenting the wrong pieces that my body has been attuned to, okay? In my fetal development, I will destroy any beta cells or any recognition to proteins that are normal. Those cells will be eliminated from my genome. And so if I present that piece of hemoglobin when I'm a baby, that protein, that cell gets destroyed, and I will never recognize it again. But if I introduce a new protein, a new organ, and I'm presenting that piece of hemoglobin, my body recognizes it as foreign, even though it is a perfectly good protein. So what you have to do is find a quote-unquote perfect match. Very rarely do we ever find a perfect match. But of those 18 different classes, you want to have as many of the 18 exactly the same as yours. because it'll take these proteins and present pieces. Yes.
What's the minimum match?
I can't remember what the minimum maximum is. Very rarely do we ever get 18. But you have to have a certain number. You can tolerate a few changes. I wouldn't hazard to guess how many, but anybody who has a transplant will also be given treatments of immunity suppressive drugs for the rest of their life. Which then, how good is that? You suppress your immunity for the rest of your life. Is that a good thing? Well, it's better than being dead because you can't have a heart, because that doesn't function. Again, it's a risk reward. Have a little bit less tolerance to being infected by viruses, but I can tolerate the response that I won't break down that new heart that I've gotten and won't allow my immunity to break those cells down and degrade the tissue of the new heart. And so you need to, this is where we found, we didn't actually know anything about this until we started doing transplants, and all of a sudden, some people's transplants took, and some people's didn't, and there was a lot more that didn't. And we saw that it wasn't just failure, but there was an attack against that. Very similar to how we discovered the blood types. We saw as we transfused blood, there were complications in certain blood types. And so we have to match those up. So when you are looking for a transplant, you get put in the system. What is the most important thing is matching up what classes you have. And that will put you higher on the list for certain organs. That is why you tend to, if you have relatives that can donate a organ like kidneys, you can function on one, you have two, you usually try to test with your family because there's a higher likelihood they would have a combination. Although that's not a guarantee, but there's a higher likelihood that they would have the same combination.